Chapter 8, Biodiversity. This chapter is going to focus on microscopic organisms. In this chapter, we'll talk about the origin of life, an overview of prokaryotes, and then we'll talk about the two domains, archaea and bacteria. And then we'll talk about, since bacteria asexually reproduce, then how can they have new genetic combinations? So we'll talk about the transfer of DNA among bacterial cells. We'll also talk about the evolution of eukaryotes, and that will include the first eukaryotes, which are protists. We'll then talk about the origin of multicellular life, and then we'll step back a little bit and talk about viruses and non-living parasites, such as HIV, prions, and viroids. So as an opening question, how did life arise? Imagine you're in a time machine that takes you back in time to early Earth. Describe what the first life on Earth might have looked like, and what life might be similar then that is different in life on Earth today. So the one thing that always mystifies me for some reason is that when Earth formed, there was no oxygen. There was water vapor, nitrogen gas, methane. There were all different kinds of gases, but there was no oxygen. And I always think that that's so amazing. So the first organisms did not use oxygen for respiration. Life on Earth likely appeared about 3.9 million years ago. Early Earth was not very hospitable. Though we don't know for certain how the Earth formed, there's a Big Bang theory. Scientists hypothesize still what may have occurred. Biologists hypothesize that life originated in a series of stages. So what were the first living cells like, and where did they arise? One hypothesis is that the unique conditions of primordial Earth fostered biogenesis, that is, the formation of new living organisms. In 1953, Urey and Miller demonstrated that organic monomers can form under conditions that simulate early Earth. So this contraption here of glass bottles and rods sort of looks like the early Frankenstein movies, but the upper bulb with the electrodes in it is the atmosphere with lightning, and the lower bulb represents the oceans where water is evaporating and condensing and coming down. So they prove that with basic, very simple molecules, that nucleotides, amino acids, sugars, all the basic building blocks of the macromolecules could form. Early Earth's extreme conditions may have bonded monomers into organic polymers. So remember, these organic polymers are getting to be larger and larger molecules. A key stage in the origin of life was the formation of duplicating molecules. So what do I mean by duplicating? That means that they had building blocks. These had building blocks, and then these building blocks could be connected and form really large macromolecule. Life depends on a molecule that can copy itself in order to pass down information. And that molecule, believe it or not, is RNA. You might have thought DNA, but many scientists now believe that it was an RNA world first before DNA came along. And the reason is that RNA carries information that makes an organism that organism, but it also has catalytic capabilities. It is a more actively, chemically active molecule than DNA, which is very stable. Cell membranes may have formed as lipids that spontaneously came together like oil droplets in water. So it's sort of amazing to think what was the probability of RNA entering inside this oil droplet and that being the prototype of the first cell. Another opening question for section 8.2 is, what do you know about prokaryotes? So there's two types of cells, right? Prokaryotes and eukaryotes. So pro means before and kary means kernel, prokaryotes. That kernel represents the nuclear membrane. So before a nuclear membrane, there were bacteria. So bacteria do not have nuclear membranes, and they're usually very small. So all plants and animals and fungi, those are eukaryotic cells. In other words, they have a nuclear membrane. They have a true kernel. So prokaryotes do not. So what do you guys know about prokaryotes? I'm thinking in my mind, what are three things that I know? They're small, they're ubiquitous, and they're ancient. Prokaryotes were the first groups of life to evolve on Earth. So we can say that prokaryotes have relatively small, simple cells, and maybe for about almost the first 2 billion years, they were the dominant form of life on planet Earth. Prokaryotes are ubiquitous, and today they're found everywhere there is life, including many places where other organisms can't survive. So one of the places I'd love to go to is Yellowstone National Park and see Old Faithful, the geyser. So there are archaeans that are living in the hot water of that geyser. So the two domains, as I mentioned before, that have prokaryotic cells are domain bacteria and domain archaea. Both typically have unicellular organisms. For bacteria, they're ubiquitous, they're found everywhere, and some can cause disease and some are very beneficial. When we think of domain archaea, archaea means like archaic, old and ancient. Most of them are unicellular, but another characteristic about them is that they love extreme environments, extremely salty, extremely acidic, extreme high pressure, extremely cold, extremely hot. Prokaryotes have unique cellular structures and properties. For sure, all prokaryotes have a cell wall, and this provides protection. Many also have flagella. So flagella is a long hair-like structure, and they can have one or more. So a flagellum is one, and flagella means many. But this helps them move towards food and maybe away from danger. And many prokaryotes also have a sticky capsule. This capsule surrounds them and it provides protection and allows the cells to stick to surfaces. Some prokaryote species can form what we call an endospore. It's sort of a very thick shelled protective container, sort of like maybe forming a seed. And then when conditions get better, then the bacteria resumes its function. It's awakened from its hibernation inside of this seed. Okay, or inside of the spore. Many prokaryotes reproduce by binary fission. And to me, binary fission is what I call bacteria mitosis. So this is where the circular chromosome replicates, moves to opposite poles, and then the cells pinch off, forming two daughter cells. Prokaryotes exhibit a wide range of shapes and structures. So one of the three major shapes are cocci, bacilli, and the spiral forms. Cocci are like little spheres. You can see them as single cells, or you can see them clustered in groups of 4 or in long chains. Bacilli are rods, and sometimes under the microscope when you're looking at them, they seem to me to look like eyelashes. They're sort of long and they sort of have a little bit of a curve. And then the last form is the spiral. And you can imagine that one is curved and it has a corkscrew type of shape to it. Prokaryotes also exhibit a wide range of nutritional habits. So in addition to photosynthesis and the consumption of other organisms, many prokaryotes can even produce their own food directly from the environment. So they're very inventive, and they can use different terminal electron acceptors or energy sources. So that's why you can see that bacteria can easily live deep in the oceans because they're using different sources of energy. Prokaryotes live together, and they can form unique features. And one of these features that they form when they live together is called a biofilm. And you guys have seen several biofilms, so I'm going to give you some examples of a biofilm. And in this picture here, the major one is the plaque that is found on your gum line when you eat, and if you don't brush your teeth, you have a biofilm, and that is called plaque. And that means colonies of bacteria are living in this sugary matrix that is sticking to your gum line and to your teeth. But you can find scum on the top of like ponds, and that's another form of biofilm. You can also find biofilm on crusts on the surface of soils also. So there's a lot of places where you can see that bacteria are congregating together, and they're in this sugary matrix that's sticking on to the surface. So imagine we were able to send a probe to another planet. The conditions on this planet, while similar to Earth, are much harsher. Temperatures swing from high to low, and oxygen levels are low. So some of the questions are, is it worth looking for life in such an extreme condition? Can life even survive in these conditions? And explain your answer. So what do you think? I sort of think that it's worth exploring, and if nothing else, evolution tells us life always finds a way to survive. Life always finds a way to survive. Prokaryotes in the domain Archaea are often found in extreme habitats. So some examples will include methanogens, which produce methane gas, halophiles that like salt, and thermophiles that like heat. So salt lovers, you'll find them in oceans or like the Dead Sea or in a lake that the waters evaporated, leaving a lot of minerals behind. And thermophiles are found like Old Faithful, the geyser. So in this example here, in this slide, we have Africa's Lake Natron is perhaps the world's most caustic lake. The orange color is due to the red pigments found in salt-loving Archaea. So actually it's giving the lake its color as the bacteria that lives there and loves the salt. Methanogens emit methane, and if you look at the word methanogen, like Genesis, it's the beginning of it, so they're synthesizing methane. And this can be found in several places. It can be found in swamps and bogs, but another likely place is that it's found within landfills that have nearly oxygen-free conditions. Halophiles survive in salty environments, so they can tolerate salt concentrations 5 to 10 times higher than that of seawater. Thermophiles love the heat, and they can live in areas where temperatures are boiling. And so this is, in this picture here, it's the Grand Prismatic Spring at Yellowstone National Park. So you can see all the colors, and this is due to the bacteria that are living there. Okay, so the opening questions for 8.4 are true and false. True or false? Are there more bacterial cells in your body than there are human cells? True or false? There are more bacteria in your intestine than there are people on Earth. True or false? Bacteria can produce oxygen for us to breathe. So I think the answers to all of these must be true. Did you get it right? Prokaryotes in the domain bacteria are very numerous and common. Bacteria thrive in most of the Earth's habitat, and that includes in and on our bodies. So we're like little walking islands that contain all of these bacteria, and there are actually more bacterial cells than there are cells of ourselves. Bacteria can be harmful or helpful. And each bacterium is microscopic, but their collective impact is enormous. So most organic carbon here on Earth, is in the form of things that we can't even see with our naked eye, which are the bacteria. And I think that that's awesome and phenomenal. Helpful bacteria have a positive impact on ecosystems. So when we think of our atmosphere right now, it's not 100% oxygen. Actually, it's mainly nitrogen gas, as in this diagram here. But the nitrogen gas can't be used in the form of N2 by life. But there's very few bacteria, and these are called nitrogen fixers. And they can actually take the nitrogen gas and convert it into something like ammonia. Now, unfortunately, the picture of me is covering NH4, which represents ammonia, but ammonia is a form in which the plant can absorb and use. So this is so critically important. Bacteria also are great decomposers, and then when they decompose an organism that lived before, they're also releasing a lot of nitrogen back into the environment. Bacteria are important in sewage treatment. So microbes are used all the time to actually decompose all of the waste that is flushed to the wastewater treatment plants. Another place where they're critically important is for bioremediation. Bacteria can be introduced or they can be fertilized so that they multiply in numbers to actually remove pollutants from a specific environment. And this is an awesome field of study. Bacterial infections account for about half of all human diseases. So pathogens are really important, and there are relatively few species of bacteria that actually cause serious illness. And so here you see different kinds. You have Staphylococcus aureus. This is like the MRSA, Bacillus anthracis, Salmonella, and then Lyme disease. So there's all different kinds, and they can cause a lot of ill health. Opening questions, true or false? Okay, true or false? Does bacteria reproduce asexually? Yeah, that's right, it's true. Soap and water are more effective at killing germs than antibacterial and sanitizer. That one I'm really not that sure about. And let's look at the last one. The only good bacteria is a dead bacterium. So what do you think about that statement? If you're too clean at home, I think that causes problems too, because then when you're in touch with the general world that is teeming with bacteria, you could definitely get sick. Bacteria have one chromosome and reproduce asexually. So when you see the word asexually, A in front of the word, that prefix means no. There's no sexual reproduction. So what is sexual reproduction? Sexual reproduction is new genetic combinations. And so binary fission is what I call bacteria mitosis, and it results in two identical cells. So you start off with one cell and it divides into two, and that's it. That is called actually binary fission. And as a result, bacteria in a colony are genetically identical to each other. But this doesn't mean that bacteria lack ways to reproduce and utilize these new genetic combinations. Okay, so it says here, although they reproduce asexually, bacteria can transfer DNA between cells. Interestingly enough, a side note is that I worked with a man who actually looked at genetic diversity in bacteria, different types of bacteria species. And I think that it's a very interesting work. And I think he wanted to answer what makes a bacteria cell become a pathogen, like what turns on the switch. And so there's different ways that bacteria can in fact create genetic diversity by transferring DNA between cells. And the way that they can do this is through transformation, transduction, conjugation, and plasmid transfer. Dead bacteria can release DNA into the environment, and when they do, there's foreign DNA in the environment. So then living bacterial cells can actually take them into their own cells and go through a transformation. They'll take that foreign DNA that was released by dead bacteria and incorporate it into their own chromosome. So this is called transformation, and this creates new genetic combinations. Another way of DNA transfer between bacteria is through a bacteriophage. That is some type of virus that is infecting a bacteria. And when it does, it's inserting genetic material. And this genetic material is actually, they can be carrying their own viral chromosome segments, or they can be carrying fragmented bacterial chromosomes. And these are injected into one bacterial cell into another. And this kind of transfer of DNA through a virus is called transduction. And this virus that infects bacteria cells is called a bacteriophage. The next way that there's a transfer of DNA is through forming a physical bridge. And this physical bridge is called a mating bridge that is made by sex pili. Sex pili are little hairs that actually hold one cell to each other, to another. And so there's a donor cell that will actually give a piece of DNA, and it will give it to the recipient cell through this mating bridge. And so we have genetic material being transferred from one cell to the other. I don't consider this true sex, because in true sex you need gametes and you need meiosis. Plasmids can also be transferred. So the question is what is a plasmid? We all know that every bacterial cell has one circular chromosome, but somewhere off to the side are small circular chromosomes, and these are called plasmids, and they act independent of actually the cell plasmid. These plasmids, or little small circular pieces of DNA, can come from the environment, and they can also be transferred from one bacterial cell into another. So how did eukaryotes evolve from prokaryotes? Prokaryotes were the only cell type on Earth for almost, it says here 1.4 billion years, but I always say to myself 2 billion years. And then eukaryotes evolved about 2 billion years ago. So what made them different from bacterial cells? Do you have any ideas? Well, one is that a eukaryote has an endomembrane system. An endomembrane system consists of two main components: interconnected internal membranes, or membrane-enclosed organelles. And so this happened by the infolding of the plasma membrane. And by doing this, a lot of structures began to form within the cell. So if we look at an ancestral prokaryote, we see that it has a plasma membrane, DNA, and a cytoplasm. There's no nuclear membrane. Then the plasma membrane begins to infold. It begins to implode. And when it does that, it starts creating all these curves and crannies. And then before you know it, you have the birth of the nuclear envelope and the endoplasmic reticulum. So the nuclear envelope protects the nucleus and the chromosome, and then the endoplasmic reticulum helps with actually either lipid synthesis or protein synthesis. Endosymbiosis, another way that prokaryotes are different from eukaryotes, is that there's other organelles within a eukaryotic cell. How did they form? If they didn't form through the infolding of the membrane, then maybe it was through something else called endosymbiosis. Endosymbiosis is when one species lives inside another host species. So the idea is that some of the organisms or organelles that we see inside of the cell, their birth comes from another species that was engulfed but didn't die and stayed alive. So one idea is that a huge cell actually engulfed an oxygen metabolizing bacterium. And this cell eventually became the mitochondria of the eukaryote. The other thing that a cell could have engulfed is like an algae cell, and the algae cell didn't die but instead lived. And this is the prototype for actually algae. So algae can live in animal cells. So what are some of the unique characteristics of the animal, plant, and fungal kingdom, which are all eukaryotes? So if we move away from prokaryotes to eukaryotes, what makes these cells so unique? So the default kingdom in this group is called kingdom Protista. And this is used to describe the earliest of all eukaryotic cells that likely evolved 2.1 billion years ago. This is phenomenal. And so they're not a plant, they're not an animal, and they're not a fungus. But I will say this, some are plant-like, some of them are animal-like, and some of them are very fungal-like. We believe that protists evolved around 2.1 billion years ago. Most are unicellular, but many are multicellular. And the multicellular ones are the ones that are the algae, the plant-like ones. Protozoans are little animals. Proto meaning before, and zoans, animals. So they're in the kingdom Protista, which is the first kingdom of eukaryotes, and they attain their nutrients primarily by eating. So some of these, like Paramecium, have cilia all over, little hair-like structures all over the cell. Giardia is an intestinal parasite that has flagella, and then we have Plasmodium, which causes malaria in a red blood cell, and actually the different stages of the life cycle are carried by the mosquito to other hosts. Amoebas are also a protist. They're a little animal, and the margins, like you see the amoeba in the upper right-hand corner, they move around and they capture their food. And slime molds are protists that resemble fungus in appearance and in lifestyle. And sometimes they can be in their single cell stage, which is definitely going to be an organism that looks like the amoeba. Protists are very diverse. So, for example, algae. Algae are photosynthetic protists able to produce their own food from sunlight. And we can see algae on land, and we can see forests of seaweed under the surface of the ocean. What do you see as major advantages and disadvantages of a multicellular organism versus a unicellular organism? I can think of some. I think that when you're big, you're not as affected by the environment as you are when you're really tiny and microscopic. So that would be one advantage or disadvantage, depending on how you see it. When you're only a single cell, you don't need many resources, but as a multicellular organism, you have to have a lot of resources to stay alive. The origin of multicellular life was a major milestone in the evolution of life. Biologists hypothesized a series of steps that may have occurred in the evolution of multicellularity. So if you look at this diagram, you see we have a single cell organism with one flagellum, and then it starts living a colonial lifestyle. And then in that colonial lifestyle, some of the cells start to specialize, and that's why you see, like grapes with tails on them versus like white cells at the bottom. So some cells are used for movement, other cells are used for ingestion. And then lastly, you see the formation of gametes, the formation of sex cells that will make those organisms true to their species. For almost the first 2 billion years of life, life was unicellular. So here we see that life was unicellular, and that the origin of eukaryotes is thought to have occurred around 2.1 billion years ago. In the last part of the geologic timescale, you see the evolution of jellyfish, mushrooms, plants, and then man appeared 2.5 million years ago. So this would be the most recent addition. So now I'd like to step back. We've talked about multicellular life. Now I want to talk about ultra microscopic life, and those are the viruses. So are they alive? So many people think that they're not alive, and that would include the author of this book, Dr. Simon, but I think many are now thinking that we can consider them to be alive. And it's because they need to have the characteristics of life itself, which are reproduction, growth and development, energy use, order, and to be composed of cells. So do they reproduce? Yes. Do they grow and develop? Maybe not really. Do they produce their own energy? Not really. They take it from others. And by the way, they can't reproduce unless they have a host cell. But there is order, and I don't know if you would actually consider a virus particle a true cell. A virus has no cells and can't reproduce on its own, but a virus must infect a living cell and direct that cell's internal machinery to make more viruses. So viruses can definitely not reproduce on their own. Viruses are very simple in structure. They're much more simple than a cell. So they usually have one or more recognition spikes. They have some kind of nucleic acid in the inside, which could be DNA or RNA, and they have a capsid. This capsid is the shell, and it's made out of proteins. So a virus in its cycle of life has two types, a lytic cycle or a lysogenic cycle. In a lytic cycle, viral DNA replicates using the cell's machinery, the cells of its host, and this culminates in the death of the host cell. In a lysogenic cycle, viral DNA is inserted into the bacterial chromosome, and the virus remains dormant for long periods of time. So here you see the difference between a lytic cycle and a lysogenic cycle. So on the left-hand side with the lytic cycle, you see the infection of a virus and that it hijacks the internal machinery of a cell to make little more viral particles which burst out of the cell. In a lysogenic cycle, actually the DNA or the genetic material is incorporated into the genetic material of the bacterial cell. Viruses are capable of infecting a broad variety of animals and plants. So in this slide you see influenza virus, and this looks like the Spanish influenza of 1918, I think. And then on the other side we see leaf damage caused by the tobacco mosaic virus. Okay, we have another opening question. What do you know about HIV? What are at least 3 things that you know about HIV or AIDS? I know it's devastating. I know that it can infect a lot of people, and I know that the mode of transmission has to be some kind of bodily fluid. Okay, so HIV is a virus that cripples the human immune system. And when that happens, we call this AIDS, acquired immunodeficiency syndrome. And this is caused by HIV, the immunodeficiency virus. HIV is a retrovirus, and it has an RNA genome. And so it targets the cells of the immune system, and it spreads by sexual contact. Now, HIV uses reverse transcriptase to convert RNA into DNA, and then once that is done, then HIV essentially hijacks the cell's own DNA expression system. So here in 8.10, we see the HIV life cycle. And this huge cell here is a human helper T cell, which is part of the immune system. So here you see the HIV virus actually at the top, and it's injecting RNA, which is converted into DNA. And once that happens, it's creating viral particles that will eventually burst from the cell. And when this happens, the cell dies and the viral particles infect other cells. Can a virus evolve? A virus is not alive, but it does undergo a process of natural selection. So, when you take the flu shot every year, a group of scientists get together worldwide and they decide what will be the composition of strains that will affect human beings. And then based on what they think is their best prediction, then they make they prepare the solutions for the flu shot. A pathogen is any agent that causes disease. So living organisms such as bacteria and fungus and proteins can cause disease, just like viruses can. Non-living viruses are also pathogens. There are two other types of pathogens that are even smaller than viruses, and these are prions and viroids. A prion is an infectious protein, and it is a misshaped or misfolded protein. And the bad part about it is it gets other proteins to actually misfold also. So prions are capable of clustering together and disrupting brain function. So this is one terrible thing that can happen from having prions. Viroids, they're very small circular RNA molecules that can infect plants. And they don't encode proteins, but can replicate in host plant cells by using the host's own cellular enzymes. And here they give an example of a potato that's been infected with a viroid, and this is what's causing the cracking.